A very good afternoon to all of you. Myself, Dr. Faria Fatma, and I'm going to teach you a new topic related to the biochemistry, that is cell division. Cell division is basically the division of a cell. Here it is a picture of animal cell as well as plant cell. These both cells are basically eukaryote in nature. Cells are the basic unit of all living organisms, and in every hour, about 1 billion cells die and 1 billion cells are made up in our body continuously. In animals or humans, they make bones, muscles, skin, and blood, while in plants, they continuously make leaves, roots, stem, and flowers. As the organism grows, the cells must reproduce. Cells must also divide because old cells die and need new cells to replace them. These are the structures of the nucleus where the nuclear membrane is covering the nucleus containing the chromosomes. Chromosomes are made up of DNA, and that DNA contains a portion of genes that instruct the proteins for our important physiological functions. Chromosomal structure. Chromosomes are basically those structures that contain genetic information and made up of DNA and protein. The genes are the small stretch of DNA that code for a particular protein. Centromere, that is the central part or point where the chromatids are joined. The chromatids or chromosome contain identical parts. Strands composed of chromatin makes the chromosome. The chromatin is the material that make up the chromatids and composed of DNA and protein. So it is a simple structure that resembles the morphology of chromosome. In humans, there are 23 pairs of chromosomes, one from man and one from women. These are known as homologous chromosomes. So let's move to cell cycle or cell division. The cell division is composed of the division and the cell cycle. Cell cycle is basically the regular sequence of growth and division that eukaryotic cells undergo, while the prokaryotic cells undergo binary division. The cell cycle is divided into three main stages: interphase, mitosis, and cytokinesis. In interphase, the cell grows into its mature size, makes a copy of its DNA, and prepares for division. In mitosis, one copy of this DNA is distributed into the daughter cell. In cytokinesis, the cytoplasm divides and cellular organelles are distributed into the two cells, forming two cells or four cells from the parent cell. This is the cell cycle where all the sequences of cell division takes place. It is composed of G1 phase, S phase, G2 phase, and M phase. G1 phase refers to growth phase, S phase refers to synthesis phase, G2 phase represents second phase of growth, and M phase again represents the division phase where the cell division is going on. The first phase, that's interphase, where the DNA replicates, ensuring that when it divides at the end of the mitotic phase, each of the newly created daughter cells would contain a full set of genes. The interphase is again divided into three separate parts: G1, S, and G2. Interphase is that stage of cell where it occurs most of its life, and it is one of the longer phases in the cell cycle. Here, the cell chromatin condenses into chromosome. The chromosome looks like an X-like structure where each chromosome is made up of two identical sister chromatids attached by a centromere. This is again created in the S phase of interphase. In interphase, the G1 represents the cell growth phase where the cell doubles in size. It is basically the period of growth for a cell. During this phase, new proteins and organelles are produced. Cells produces all the structures it needs to carry out its function. Second phase is S phase, where it is known as DNA replication or synthesis phase. The cell makes an entire copy of the DNA of the cell. This happens because the new cells also need a proportion of DNA for functioning and survival. Think of this phase as placing the DNA on a copy machine, and it makes the photocopy of its own DNA. The third phase is the G2 phase, where the cell produces the organelles and structures that are needed for the cell division. The G2 phase is the shortest part of the interphase cell, and it prepares to divide. The cell produces structures needed for cell division. Now it goes on to the next phase, that is the mitosis phase. During mitosis, the cell's copied genetic material separates. The cell prepares to split into two daughter cells. This allows the cell genetic material to pass into the new cells, and the resulting daughter cells are genetically identical. There are again various substages of mitosis, that is, prophase, metaphase, anaphase, and telophase. In the prophase, the nucleus disappears. As you can see that the nuclear envelope is disappearing. The spindle fibers formed in the cytoplasm. The spindle fibers attaches to the sister chromatids. They are preparing for division. In the metaphase stage, the sister chromatids are pulled to the center of the cell. They line up the cell at the equator plane. In anaphase, the spindle fibers begins to shorten. The sister chromatins are pulled at the opposite ends of the cell. That increases the separation of the spindle fiber, while the polar microtubule help in attachment of the centromere chromatid. In the telophase, approximately 80% of the nuclear DNA has been separated at opposite poles of the cells and begin to unravel, and new nucleolus begin to form. So in short, there are four stages of the mitosis. Just remember PMAT. P stands for prophase, M stands for Mphase, A stands for anaphase, and T for telophase. Again reasoning: prophase, condensation; Mphase, means equatorial plate; anaphase, the spindle fibers are coming opposite poles; and in the telophase, the nucleus is formed containing the chromosome, and whole cells divide with the help of cytokinesis. Last but not least, cytokinesis is basically the division of the cytoplasm that results in two separate daughter cells with the identical nuclei. Sometimes the cell cycle regulates how long a cell lives. Sometimes cells die because they have suffered injury or damage that cannot be repaired. One more thing, apoptosis. Apoptosis is a condition where a cell also dies when it's no longer needed or non-useful to the organism. This is basically the controlled or regulated cell death. The cell on the bottom is healthy and normal, which cell on top is undergoing apoptosis. With this slide, I would like to thank you all for listening so patiently. We will meet you again